Let's take a look at the pancreas. So the pancreas is both an exocrine and an endocrine organ. Exocrine because about 99% of its cells are acinar cells. And the acinar cells are out here. I'm circling them in just a few places. These cells produce digestive enzymes that are then going to go into the duodenum of the small intestine. And they also produce a pancreatic juice or bicarbonate alkaline juice that helps to neutralize chyme that's come from the stomach, because the enzymes that come from the acinar cells and several others in the small intestine require an alkaline environment. Now let's look at the endocrine part of the pancreas. The other 1% is found in the islets of Langerhans. So the islets of Langerhans make up about 1% of the pancreas, and they contain a couple of different cell populations. The alpha cells that produce glucagon, that's about 20% of the cells. The beta cells that produce insulin. Delta cells make up about 4% or so of the islets. They produce somatostatin, which is a hormone really similar in function to GHIH that's produced by the hypothalamus. And somatostatin here limits or stops the production of both insulin and glucagon. And then a fourth cell population that is sometimes called PP cells or F cells. They secrete pancreatic polypeptide hormone. So in reading some of the scientific literature, you'll see that PP seems to have an effect on weight in certain populations of humans and on appetite. It's also been found to be pretty low in people who exhibit Prader-Willi syndrome, which shows hyperphagia, so people that seem to eat uncontrollably, like not be able to stop themselves from eating. But we're going to focus on glucagon and insulin. Now, in some histology slides, you'd actually be able to tell the difference between alpha cells and beta cells in the islets of Langerhans. You can't really do that in these slides very well, but you should know the differences between the hormones they produce and then what glucagon and insulin do, because they're pretty powerful hormones. And insulin disorders are part of the features of diabetes mellitus. So let's look at glucagon first. Its overall effect is to increase serum or blood glucose levels. And it does that by stimulating glycogenolysis, which is the breakdown of glycogen, that polymer, to individual glucose units in the liver, and then that is exported out to the blood. It also stimulates gluconeogenesis, and that uses intermediates from amino acids or fatty acids to make glucose-like products or intermediates that can then be used in the Krebs cycle to make ATP most effectively. It also stimulates the process of lipolysis in the adipocytes, which would be breaking down that stored triglyceride into fatty acids that can then be used to go into the Krebs cycle and extract more ATP than you can get from a glucose molecule. The other cell population that we'll look at in the islets of Langerhans are the beta cells that produce insulin. And the overall job of insulin is to decrease serum glucose or to decrease blood glucose. And it does that by telling cells, particularly the ones in the liver and the adipocytes, to take up glucose from the blood. So it increases transport of glucose from the blood into those cells, and then those cells can store that glucose as glycogen. It also inhibits gluconeogenesis. So where glucagon stimulated that process, insulin inhibits that process. And although gluconeogenesis is inhibited in the cells, they're still going to uptake amino acids and fatty acids. Adipocytes are stimulated to perform a higher level of lipogenesis, so that's storing triglycerides for long-term energy storage, and other cells that are targeted by insulin will also increase their rate of ATP production. So they'll use the stored glucose they have to generate ATP and store that ATP, but they're also pulling in more glucose.